Welcome to Chapter 2, where we will be discussing the very beginnings of the human lifespan, including evolutionary origins, genetics, as well as prenatal development, and also the first years after birth. As you can see, some of the basic questions we start with will be the origin of our behaviors. So it's a classic nature-nurture question, but here specifically we're talking about some of our behaviors and preferences that might be caused by our evolution and programmed in some ways in our genes, and where are the boundaries of that. And also we'll stop in for the cognitive developments, the social developments, and many aspects of physical development, especially prenatal and post-birth. All that and more to come. We'll start with our discussion of evolutionary psychology. The basic idea is that many of our behaviors, we think, have some origin in terms of our evolutionary past. And some of them have evolved so that we can be more adaptive, so that we can be more likely to procreate. And a lot of them, as we'll find out, aren't necessarily the way we think about our behaviors and plan things. They're more feelings and unconscious pushes or urges. And that's a lot of what evolutionary psychology involves. We can also talk about things such as nature-nurture. Well, if we see, for example, with handedness, being left-handed, or identifying as gay or homosexual, if we find that a certain percentage, usually about 10%, for example, with handedness, maybe similar estimates, I'm less certain about the consistency, but there's a fair amount of consistency that we see both of these things in pretty much any culture across the world. And when we see that, it is evidence of some sort of genetic commonality. So we say there's got to be some genetic or evolutionary component to both of these concepts. Another example for evolutionary psychology is the drive to procreate, the drive to have a social environment, and then the decisions about who are we attracted to, who are we trustful of, who do we learn to fear and have maybe an implicit or somewhat unconscious respect for. So here are some of the dimensions or traits of these kinds of things that either pull us toward or push us away from a person. And we manipulate certain things such as skin color. You can see the amount of teeth that they show in a smile, the angular level of the chin, for example, how far and angry are the eyebrows, another thing. And so we find that typically across cultures, and again, this is an evolutionary argument, that features like this are viewed as youthful. Features such as this are most approachable, and this makes sense. A baby, if it has some sort of program to be more trusting of somebody like this compared to this, maybe they'll get treated better on average. And so those are the kinds of things that the evolutionary argument makes in terms of our behavior. Here we see some of the major contributors for the field of evolutionary psychology. This is David Buss. He's written a lot and done a lot of the theory work in terms of what is the evidence for examples of clear evolutionary impact. And David Buss has basically had a lot of controversy because a lot of people, especially in terms of gender differences, which seem to be controversial, especially he's made a lot of enemies in the feminist field, because he would say things such as sexual preference and features that drive either toward or away from approaching someone with potentially the unconscious goal of mating and procreating. Most people want to think I have conscious control of all that, and to say that some of my preferences are evolved and shared with others of my gender is somehow cheapening it. And I don't think that's what he's trying to say, but I would also agree that sometimes he could have said it a little bit more cleverly. Also, we have here Robert Plommer, excuse me. And unlike David Buss, who's looking at the theories and patterns of development, Plommon's the guy who is actually doing the gene work. He's breaking down the genome and looking at specific genes or clusters of genes. And he's a pioneer, of course, in the field of behavioral genetics, which we'll talk about in just a little bit. It's hard work. It relatively, it's newer work because we have the technology that we didn't 50 years ago, but two similar perspectives, but different methodologies to make assumptions or conclusions about consistency and patterns in human thinking and behavior. So a couple things to think about. If we look at, again, basic nature-nurture considerations, we could talk for weeks or years. So we just have to pick some examples. One thing is that oftentimes we look at very obvious differences, and we have our classic racial ethnic status, gender, age, and we look at someone who is different from us in those things, and then we assume they're different from us on all sorts of things, a range of other traits and preferences. The thing, when we do that though, and we can think about maybe a less controversial example, Ohio State, Michigan, we might think that Michigan and Ohio State football players have a lot that separates them, that makes them different. But in fact, there's just as much difference between your average Ohio State players compared to your average differences between any random Ohio State player compared to a Michigan player, right? We over-assume differences between groups from usually just a limited set of obvious differences. And we have to appreciate when we make that mistake. So this is kind of the origin and the argument against stereotypes, especially stereotypes based on very simple physical features, for example. Another thing to think about is how fast can evolutionary psychology work? Well, if we look at the 1960s, Americans have doubled income, and there are a lot of other good things suggesting that we, at least up until the 2000s, have seen a lot of progress. On the other hand, though, given this environmental positive prediction, we've seen a lot of negative social outcomes, such as divorce rates, increased teen suicide rates, increased prison population, and these are all evidence of environmental, not genetic changes. Our genes, our evolution doesn't happen anywhere near close to that limited, small timeline. So as we introduce some of our original terminology for this chapter, first it's important to think about, if we're talking about nature, nurture, what is nurture? Well, nurture is the environment, and it's very, very broad because it's every non-genetic influence. And so that could even start from pre-birth, prenatal factors, and then of course to all the things that we might be differently exposed to from the food we eat, the places we live, the education that we have, the places we work, on and on and on. We also have the idea that our genes interact with our environment. You don't have to necessarily know these for exam purposes, but in some ways our genes can sort of passively influence our environment, which means we have our genes and they don't really, when they're put in a certain environment, they don't really have much of an effect. On the opposite argument, sometimes we have our genes that evoke. So if we have genes that maybe we're just lucky to be generally viewed as physically attractive, and it's nothing that we're doing that's different, but since we have attractive genes that translate into people viewing us as attractive, it evokes responses such as more attention, more assumptions of unrelated things, but people will think we're smarter if we're more attractive. A lot of psychology research has shown that. And then we also have active gene-environment correlation. So for example, if you have a gene suggesting aggression, that means you're going to behave in ways different on average to the typical person. And if you're more aggressive or more dominant, you might be more likely to get attention. You might be more likely to get the kinds of food or other things that you're seeking. Another important thing for traditional evolutionary psychology research is then the non-shared environment. So let's say for siblings, and especially identical twins. Identical twins are, as it's been said many times, nature's control, because you're controlling for genes because identical twins have the same genes. And so any aspect in which they're different means it has to be the environment. And then you look at, well, if they grow up in the same house and they go to the same school, then what are the differences in the environment? So for example, I thought, well, maybe one plays football, the other chooses band. And then you can see the differences in maybe the career they choose, or for example, their level of persistence. We would then be able to track that back down to some of non-shared environmental differences. And again, if it's identical twins, it's not likely to be genetic. I won't spend too much time on this basic setup, but this is a classic. Before we could look at the genome and look at specific genes and correlate things with them, we had to do twin and sibling studies. In this case, we look at not just any random pair of siblings, but siblings with one that's identified as average intelligence. This is the middle 50%. And then the other twin, or excuse me, the other sibling would either have to be above average, slightly, or highly above average intelligence, or the other twin or sibling would have to be below average, slightly, or severely. And we can see all sorts of differences between these. I think the biggest one that stands out to me is if you look at, and this is not adjusted for inflation, so this is at least a couple decades old, but we look at the mean income for the average intelligent sibling and then see how much more being in the top 10 percentile translates to, and how much less being in the bottom 10% percentile translates to. And the idea is that, well, they're siblings. Chances are that they experience very similar things growing up, similar household. We wouldn't think that many of them were fed, one was fed a healthy diet and the other was fed a terrible diet. One was forced to sleep on the floor. So many of their environmental things were the same, but for some reason their intelligence with these matched pairs were different, and you can see the effect of intelligence here. So we'll come back to that example in terms of intelligence and how it develops multiple times throughout the semester. Just to show visually the identical twin argument, so we have, for example, identical twins, mono one zygote, one egg and one sperm cell, and then that divides into two. Compare that to fraternal or dizygotic twins where you have 2 eggs, 2 sperm, and these fraternal or dizygotic twins could be the same sex or could be the opposite sex. We also see a couple twin facts. We see that identical twins are pretty rare relatively, and fraternal twins are much more common, and that rate is much higher now than it used to be compared to 30 years ago, mostly because of in vitro fertilization, in which multiple eggs are placed in the womb with the idea that at least one, but sometimes more than one, will become fertilized and become viable. But we can also see, even right after conception, you can see some nature-nurture differences, in this case specifically nurture. Identical twins, exactly the same genes. This is how it would normally take place. They share the same placenta, but you can have identical twins and separate placenta arrangements. And so potentially one of these is maybe less exposed to sound or gets crushed or sat on more. Who knows what kind of arguments you could make. But these differences in nurture can start very, very early, potentially. And it's just one example of the hundreds, thousands of non-shared environmental things that we could potentially analyze with any given set of twins. A couple other ideas regarding identical twins. Remember, these are nature's controls, and so when you see, for example, a high correspondence between, if one twin, in this case, is diagnosed with Alzheimer's, then there's a 60% chance that the identical twin will also be diagnosed in his or her lifetime. So that is clear evidence of a fair amount of genetic influence, or the hereditary factor. But also we have 40% that is not explained by the genes. And so that's 40% of Alzheimer's that is non-genetic for identical twins. Now, of course, fraternal twins, they share more genetic material than your average random two people, but there's substantial differences. And so once you take fraternal twins, again, who have some different genetic information, now that risk factor or heritable component for Alzheimer's disease has been halved. And that's a typical ratio, is that often the identical twin percentage is one thing, and then the fraternal twin percentage is often about half of that. Also, we can see identical twins reared together. Again, they have their identical genetic information, but if they share the same environment growing up, then they turn out to be more similar. So again, nature and nurture both have an effect, even in identical twins. And this is a classic example of twins separated at birth. It was a family from New York that was adopting the twins from Guadalajara, Mexico. Unfortunately, there were a lot of issues with the adoption process, and they were only able to bring one of the twins to New York. Now, another family was able to successfully adopt the other twin, and they grew up within driving distance of each other, and oftentimes people would come up and say hi to them, and they had no idea who they were because they had seen their twin. So they were reunited as late teens, early 20s, and found out that, yes, they had some differences, so one liked Chinese food and the other didn't. But they had many other similarities, such as especially their personality, how they would interact with people, and the rhythm of conversation and communication. And of course, we always talk about the Olsen twins. I grew up with them, and in many ways, people assume that they're identical because they do look very similar. But in the identical twin case, they have the exact same height, up to the 1/4 inch, and one of the Olsen twins, I forget which one, is an inch taller than the other one. So they've had a very similar environment growing up, but they're no more identical than your average brother-sister pair, fraternal twin. Now we're going to start talking about genetics. We've talked about some evolutionary things, some twin studies. Now we know that some things do travel or are passed down in genes. These are common birth defects, or at least historically important birth defects. And what's interesting is we're not quite to behavioral psychology yet, because we're talking about, in this case, genes, but usually one gene. So with one gene, and if you know whether it's a dominant or recessive gene, and you know whether the mom or the dad had it or not, you can say for certainty whether the child will have that. So obviously these things are going to be important to know for prenatal counseling and those kinds of issues. And of course it's controversial. If you know for example, that you will pass on Huntington's disease to your child, but it won't start until early adulthood, but once it turns on, you can see the difference in the reduction in cortical tissue that corresponds to a lot of behavioral and cognitive deficits that will ultimately claim the person's life. Some you see start very early, sickle cell anemia, Tay-Sachs disease, but others like Huntington's disease has a very long onset, and cystic fibrosis is onset until early childhood. So you get a few perfectly healthy years, at least with respect to this. So the basic idea here is that there's two processes at work as far as we understand. There's one that determines the genes. So do you have the genes or not? And if you do have the genes, you're going to have it at some point. But then that at some point is the other factor. Why don't, if you have the genes, they express themselves right from conception? And that's the question. And that also, it so complicates things, but it also opens the door for positive thinking, because maybe, for example, why wasn't Huntington's disease expressed until early 20s? Well, if it wasn't, there might be a mechanism that we can keep it from ever being expressed. And so that's the logic, I think, behind a lot of potentially tragic genetic birth defects. We won't spend a lot of time on the specifics of DNA and genes, but I also wanted to talk about chromosomes, because our chromosomes contain our genes. So you zoom out from genes, and now we talk about chromosomes. You can also see here with the sex chromosomes, typically women are XX, men are XY. You can have extra chromosomes, and in most cases, just like brain size as well, bigger or more is not better. What you want is normal or average as far as a lot of biological things. You can also see that sex chromosomes disorders are fairly rare and also more often male. And so again, when you see these extra or unusual combinations, they correspond behaviorally or cognitively with difficulties and challenges. And another common thing, it seems like if you have a Y chromosome, that Y chromosome just seems to break down, and so males are much more likely to experience sex chromosome disorders, and often red-green color blindness. Most males, on average, are just more colorblind than males, and that is often passed down through the sex chromosome, the Y chromosome. We can also see Down syndrome. I'm sure we all have met multiple people with Down syndrome. And again, more is not better. They have an extra 21st chromosome, and it corresponds not only to cognitive impairment and developmental disorder, but also a common phenotype. So you see that physical features develop differently. One pattern that we'll talk about throughout development is key areas of the brain and when do they need to develop. So for example, the cerebellum, responsible for coordinating automatic movements, hippocampal shrinkage, being able to put in explicit memories and then get them out or retrieve them later on. And then the prefrontal cortex, responsible for behavioral inhibition and paying attention and filtering out distractions. And I think we can see that some of those things with the people I've known with Down syndrome, you can see some of those challenges play out and then tie them to certain brain regions. The one thing to think about is that these things sort of have to develop at a certain time, and so if they're basically behind in childhood, these things are something that typically can't just grow back later on. If it doesn't develop normally when it's supposed to, it doesn't mean that people can't learn or improve later on, but it won't change their name or their brain into what we would consider a normally looking brain. So we've been talking about some of these basic patterns. Let's actually put some terms to them. So first of all, we have the idea of heritability. So this is if your identical twin has Alzheimer's, you have a 60% chance of also having Alzheimer's, or insert schizophrenia or seasonal affective disorder. I mean, all those are some range between 0 and 1 or 0 and 100%, and rarely are genes for behaviors going to have a heritability of 1, unlike things like Tay-Sachs and Huntington's disease. We also have the idea of behavioral genetics. So this is the basic pattern we've been talking about. You can link individual genes, or maybe 1 or 2 or 3 genes, to things like eye color, hair color, height, other physical facial features. But complicated things, behaviors like personality, intelligence, aggression, they do have genetic links, but they're not just 1 or 2 genes. They are going to involve multiple genes, and it takes just a lot more work, first of all, to measure these things. There's more error in measuring things like intelligence than there is in measuring somebody's eye color. So you have what we call a criterion problem, but you also have to do a lot more correlations with multiple genes, sometimes dozens, or maybe in the case of intelligence, hundreds. So behavioral genetics is much more difficult to do, based on my understanding, than your early genetics findings. So for example, this is Steven Pinker, a notable cognitive psychologist, has done a lot of stuff in terms of child development and evolution. He was one of the first that I saw who had his genome mapped, and he saw a whole bunch of things about his genes that suggested, since you have this gene, and one was, for example, a super sensitivity to bitter taste, then we predict you won't like things like dark coffee and dark beer and Brussels sprouts. In most of those cases, they did fit with what he liked. The genes predicted something, and most of those predictions were correct. And that's the idea of niche picking. Basically, people choose things, and it feels like free will. But to some extent, many other choices, and again, depending on which one we're talking about, the heritability rate can be different, right? Somewhere in this range. But it has something to do with your genes. So for example, Japanese have often been looked at as a useful society to do genetics research because there are cultural taboos on having children who aren't also Japanese. And of course, in the support of diversity, that's not necessarily good policy, but scientifically, looking at the effects of genetics, that can be useful. And so what we find is that on average, Japanese have a set of genes that make them overly sensitive to sweet taste, and so they prefer lower sugar foods. Typical foods that we would get as United States citizens, such as ooey gooey desserts with hot fudge and whipped cream, would be perceived by them as too sweet, and so they avoid them. Now, of course, your genes do not perfectly predict your behavior, not anywhere even close. These are just general patterns. And so, for example, with Steven Pinker, he has the gene predicting that he would not like dark beer, but it turns out he does anyway. So again, we're just doing the correlation prediction game, but not saying that we understand anything perfectly outside of very limited disorders and physical features through somebody's genes. So relative to what we've already been discussing, continuing some of these basic themes, first of all, we have the idea of polygenic. So behavioral genetics, we're talking about behaviors, they're almost always going to be polygenic. If somebody is more aggressive on average than the typical person, and we can link that to genes, well, it's not just going to be, oh God, one gene, one disorder, such as another example, dimples, right? It's going to be multiple genes, sometimes dozens, sometimes hundreds. Of course, this makes it tougher to do genetic counseling. If you want to have a child who is less aggressive, well, it's hard to know, looking at their genome, if they are actually going to be more aggressive on average. And even if you did have some genetic information, you've got a dozen genes that you've linked to aggression, chances are the environment is has much more of an effect. It accounts for more of that behavior than the genes. And so, we're still in the infancy on, even once we find the genes that mostly drive a behavior, once we have enough to say it can explain 25% of a personality or IQ, then the next question is, what do we do with that, right? And so here's the example of male pattern baldness, right? There's a lot of pseudoscience here. It's like, well, we've learned a lot about genes, so we can use our understanding of genes to solve everything. Well, I don't know. Can you identify the genes for male pattern baldness? Probably. But then again, what do? One example is epigenesis. So now we've identified the genes, and we know for one thing the genes that are involved and explain a certain amount of that, or a certain percentage of the heritability. Well, we can't just change your genes. You have the genes you have, which is kind of depressing at first. But going back to the Huntington's disease example, when does it kick on or kick off? This idea of epigenesis, meaning that typically gene expression seems to be able to be turned off or turned on. We often don't know the reasons why or the mechanisms or how to do it. All we know is the evidence that it does happen. And so I think, again, we're very early in this journey of understanding these things, but it's hugely powerful to think about the idea of, let's say somebody has genes that maybe predict some negative outcomes, but if we put them in the right environment, such as, for example, a critical period for development, maybe that environment could keep those bad genes from expressing themselves. And again, we're talking very general because we're not even clear how that would work with humans. Okay, now we're going to take a step back from our discussion of genetics, and let's look at some prenatal development. Again, we could do a giant deep dive and have a whole class, or at least a month of class, devoted to this. Your book does a good job with some things that you may want to be familiar with. The classic what to expect when you're expecting, when you're in this position. Either you're carrying the baby or you're on the other side and caring for someone who is carrying a baby. Think about prenatal stages compared to trimesters, right? Trimesters are where we look at the mother's experience and divide that total 9 months into 3 equal chunks. And the first chunk is where you might have morning sickness, but the baby is very small. The second trimester is where things You can tell things are changing, but you're not very uncomfortable and can live fairly normal life. And then the third trimester, on average, that at least at some point during that, mom's going to become very uncomfortable, and the bulk of the baby's growth is happening then. Contrast that with the prenatal stages. These are not equal time lengths, not at all. They're divided instead of the mother's experience, but rather to what's happening and the boundaries of the soon-to-be baby. So, for example, the first 2 weeks are what's called the germinal stage. So, where the germ or the zygote moves from the fallopian tubes to the uterus, where that's when it can stick to the uterine wall and then develop normally. And of course, if it doesn't make it to the uterus, it can be called an ectopic pregnancy that will not be viable. We also see not much development in terms of looking like a person, but we see at least the formation of these 3 derms. So you have the ecto, or outer derm, that will become the hair and the skin. We have the meso, or middle layer, which will become the bones and skeletal system. And then we have the endoderm, the inner derm. This layer will become the internal organs, such as the lungs and heart and liver. Then we get to the embryonic stage. This is from 2 to 8 weeks, so still early in the overall pregnancy. Here's where we now see differentiation of basic organs within the endoderm. We also see this as a critical period because exposure to drugs, either intentionally or unintentionally, are very bad for the baby and most likely to cause a miscarriage or termination of the pregnancy. And then once we get to the 8 weeks in, or 2 months, now we call it a fetus. And here we start to see the development of things that we basically identify with the human experience, such as moving, growing neurons, sensing things we imagine, sensing hearing and taste. We also see cohort effects, right? So for example, if someone grew up in a time of war, so mom was pregnant with the fetus in a time of war and there were food shortages and you couldn't go outside, chances are you can trace that even if the war ends after birth. On average, that cohort that had limited nutrition, if we track them into adulthood, will have probably larger rates of cognitive disability, physical limitations potentially. And of course, drugs and alcohol and smoking are bad for the baby at any time during the prenatal stages, but the idea of teratogens, monster causing or birth defect causing, drugs and alcohol are still bad, but they're less likely to cause the elimination of the baby, but instead do long-term damage either cognitively or physically. So things like fetal alcohol syndrome, which start in the fetal period. We can see a couple general patterns that we'll have terms for later on. So we can see the relative size once we get to the fetal stage. And of course, there's this just giant acceleration of growth in the final month or two. And we also see the change in proportions, right? It looks more and more like a human as we get to birth, but still they have big heads relative to their bodies and little tiny feet and hands. And that's normal. And then a lot of the rest of the development is the head kind of stops developing or slows down drastically, and the rest of the body kind of catches up in predictable ways. We also have the idea of how do we invest in babies? Babies, we often don't think about babies as a resource, but they definitely are. A healthy fertility rate is, as we talked about with population pyramids, very important for the health of a society. And so you think everybody would be on the same page in terms of investing to make sure that as many babies come to term, but also not just come to term and survive, but have the best likely outcomes so that we make sure as many critical periods are met without any issues. And this is the case of prenatal care. So again, what to expect when you're expecting. You know, if somebody is, for example, a teenage mother or another mother who doesn't have a lot of support maybe they don't have someone that they can call anytime who's been through it before. That's when prenatal care becomes extremely important. And we look at the different percentages of expecting mothers who get prenatal care in the first trimester of pregnancy. And some states are doing much better than others. Ohio looks okay, but I think the argument should be whether it comes down to how we do healthcare, other considerations, where are they getting this in terms of Planned Parenthood, things like that. This number should be close to 100% in the United States. We can afford all expecting mothers to see a doctor or see a healthcare professional. And for example, ultrasounds, if needed, and to make sure development is happening in the normal place, and at the certain times you can see the kind of features you need to see. Also, medical family history. Are you drinking caffeine? Probably should cut that out. Or things like even if you have cats in your house, and toxoplasmosis can be found in kitty litter. You might want to find a different solution for that just while mom is carrying the child. Also, things, what am I allowed to exercise? What exercise can I do? Can I still work? How many extra calories should I eat a day? Those things aren't necessarily just obvious for every mother. And so having some sort of screening and counseling just to help mom make the best choices she can seems like a good idea to me. But again, we want these rates to be 100%, and we see that they are not in these two charts. And we see that two major factors drive getting more prenatal care. And one is being white, and also having, regardless of racial ethnic status, having more education. And again, this is another one of the things. Why am I going to college right now? Maybe the things I'm learning specifically I'm not going to use on the job. Well, we know that having a higher education is also linked to other things that maybe you're not expecting to learn in college, right? And so again, we see the difference between non-Hispanic white and Hispanic. No difference in terms of less than a high school education, but still very low percentage, under 70%. And then those rates get higher, and then the ethnic difference, racial ethnic difference, comes into play after the high school diploma level. So lots of social factors and debates that we could have if we had time. I also wanted to talk about some birth and post-birth issues. So here are some common problems that, again, you can have a child that goes through or experiences one of those things, well, the first two at least, and still comes out to be healthy. The idea, though, is that they are correlated with problems, and so let's try to avoid them if at all possible. So first of all, we have the idea of premature birth, and this is before the 37th week, so 2 months early on average. And we see that that number is 13% in the US, and it is related to afterbirth or neonatal death. And just so we're on the same page, neonate is a baby within the first 4 weeks after birth. As we'll find out, especially behaviorally, the neonate is not traditionally what we think of as a baby. It's not laughing and smiling, and it obviously is very, very vulnerable, having to be thrust in this brand new environment compared to the environment that it experienced pre-birth. Also, we have low birth weight. I don't know what the average weight is. I would guess just under 8 pounds. So under 5.5 pounds is the typical benchmark for low birth weight. Again, we see 8% in the US, half that compared to developing countries that have less access and funding for public health care. And there's some causes associated with maternal health that can impact or be related to low birth weight. And again, prenatal screening maybe can identify a whole checklist of things that are risk factors for low birth weight or premature birth, and then counsel the mother, and hopefully that counsel is taken and we get more healthy babies. We also see stillbirth. This is death 20 weeks after conception, and this is let's see, 1 out of every 200 about US births, and it seems like a low number, but compared to other countries that we often measure ourselves against, we're still behind. So I think, given especially how much we spend on healthcare, we got to get this number down. And also we have sudden infant death, and it says here, I mean, some people call it a neurological disease. I mean, if you can see post-mortem, that particular thing is rare. The actual SIDS, where the baby after birth just dies, we don't understand necessarily why. It's definitely a lot higher than 1 in 10,000. And there's some genetic links, there's some demographic variables that are correlated with SIDS, but it's still something we're still trying to understand. I think part of that might be due to education levels, and especially postnatal counseling on how to set up a crib, for example, so that the baby's less likely to suffocate. Things such as don't have your 2-week-old baby sleep with you in the bed because it is so easy to roll over and suffocate them. All those things add up and are kind of bundled in to the SIDS bucket. Now I want to talk a little bit about brain, and specifically neuronal or cortical, if we talk about just the cortex development, because there are some amazing things that are happening from conception all the way up to basically age 2 or so, toddlerhood, and then the way the brain develops changes drastically, and it stays that way for the rest of it, basically the rest of our lives. And we'll talk about that specifically in a little bit. Here we see areas associated with sensing and understanding your environment, identifying food, identifying danger, and then motor or movement coordination. And basically rats and cats, almost all of their cortex is devoted to those two things, right? Identifying food and danger, and then seeking food and avoiding danger. Now if we take humans, look at the much different ratio, and our closest animal kingdom relative from an evolutionary perspective, the chimpanzee. And we see that our brains basically have some areas that are specifically devoted to sensory perception and motor coordination, but most of the rest of the cortex is doing other things. And this is a visual example of why humans are capable of so much more higher order things than simple animals like rats and cats. We won't go into a full bio lesson, but I did want to at least show a neuron, because there are a few things that we are going to talk about. Now, one thing to keep in mind is that neurons work by connecting with other neurons. And so you've got these dendrites that are antennae and are not physically connected because they don't physically touch, but they do receive neurotransmitters from the axons of sending neurons. The other thing that's important is that a neuron can either fire or not. And when it has enough stimulation to fire, it does. Sends an electrical impulse down the axon. What covers the axon is this myelin sheath. And so unlike everything else, when a neuron is born, it's got everything here except for the myelin. And so when does that myelin form, at what age, in what areas, is hugely important to the brain becoming more efficient for all sorts of different things. Now we look at the basic pattern of synaptic and neuron density in the brain, specifically the cortex. And this pattern would even extend back before birth, but up to about 2 years, give or take, for most of the cortex. And again, not every area is going to mature at the exact same time, but the general pattern is more neurons and more connections between neurons. So you have a greater density of a network. And I think the idea here is, what does this mean? Because this, to me, represents the potential to do all sorts of things. But it also represents some limitations, because it's very dense and most of these pathways have not yet been myelinated. So we are seeing the potential for that person to represent a lot of different things, but it's not doing it in an efficient way yet. So the key thing that happens after about 2 years is that pattern switches. Instead of growing new neurons and connections between neurons, pretty much the way our brain is changing after age 2, more or less, is it is losing neurons and is losing connections between neurons. And we'll find out that that's an adaptive process because those neurons, those pathways that do stay, represent things that the child is exposed to and is doing. And those pathways are what become myelinated, which then allow the child to do those things more efficiently. Here we see, on average, different parts of the brain, and when do they reach that lowest level of what we refer to here as gray matter. So when do different areas of the cortex turn blue? And what that means is the gray matter is gone and has been replaced by white matter, which are those myelinated pathways. So we see that areas involving, going back to that rat and cat brain, for humans, areas involving visual perception, coordination of movement, and other sensory perception, those areas are basically myelinated and they're done developing, right? And what's interesting to me is we get to, let's say, age 15, most of the brain is pretty much blue, with a few exceptions here and there, except for this frontal cortex. Well, think about how a teenager is going to change in terms of how they interact with the world. And if you're thinking about a teenager as being immature in terms of morality and behavior control and logic and assessing danger, well, they can talk and walk and move just like an adult and sense the world, but they can't think about the world like an adult because those pathways just don't settle down yet, and there's still some myelination and discarding of unused pathways to do, even once we get to age 20. So if you think about this in terms of synaptic density, what areas of the brain have the most neurons and connections between neurons? Well, in terms of visual cortex development, we see the age in days, so just shy of a year. And that's the most density for vision. And then you're losing connections between neurons, you're losing neurons, and basically your visual pathways are formed right here, myelinated. Those that aren't needed are gone, right? When does that happen with the auditory cortex? A little bit later. So peak synaptic density, you have all the pathways you need to hear the world as you'll need to for an adult, and then those pathways you don't need are then eliminated. And then finally something for reasoning and self-regulation, staying focused, paying attention. Obviously, that doesn't peak probably in kids until after 3 years, right? And that's when you have all the wiring you will need, and then those pathways that are not needed are then gone. And so I guess that's important to think. Some aspect of behavioral control, and again, prefrontal cortex right behind your forehead, you should start seeing signs of clear development of that thing in your child by age 3.5. Couple other examples that we spread out. So again, basic sensory pathways get very dense and then are settled down, and then basically there's less and less development as we go. Even most of the development's done after the first year. Compare that with language, right? Tons of development that is happening, and I think this is specifically language of tracking different sounds in a given language or multiple languages. So there's a delay there compared to the sensory pathways. And of course the higher cognitive functions such as understanding math and spatial functioning, all that's going to have to be done later on. If we think about nature-nurture, we talk about maturation is a program that it's going to happen as long as the environment provides everything that's needed. So for example, we take a rat brain, and normally it's going to develop a bunch of pathways to represent at least basic abilities to deal with other rats. And we find that rats that grow up with other rats have more density than a rat raised in isolation. And then I think the idea is that for humans, all those areas that aren't devoted specifically just to moving or sensing things, a lot of our brain or cortex is responsible for being able to understand other people, being able to step in their shoes, or so to speak, walk in their path, understand what they're thinking and feeling, and it takes a lot of brain area to represent that basic ability. Another thing to think about is when we are sleeping, uh, specifically when we are in that part of sleep known as REM, rapid eye movement. This is often associated with dreaming, not exclusively, but mostly associated with dreaming, where we are consolidating memories. That is where our brain is changing in ways that help us remember things that we were exposed to recently. And if you think about being born, of course, right after birth, they're going to sleep basically 2/3 of every day, but half of that time is spent in REM sleep. And that makes sense because after you're born, you're put in this entirely new environment and you don't have any understanding of what this is. So you have to learn from a bottom-up process, as we'll say, and develop in the brain a basic understanding of faces and sounds and places and smells. And as that happens, and you can start experiencing the world from a more top-down, I know I have expectations about what the world should be, then your sleep ratio for REM sleep will be more like the adult distribution. So less than a quarter of the total night's sleep moving forward will be in REM sleep. I promise we're about done with our neuroscience for today, but just to wrap up and spring us forward into what will also continue on in future chapters, I wanted to first point out this idea of cell death or synaptic pruning. So this is that huge process that I spent a long time talking about, that basically up to age 2, we are building more neurons and more connections between neurons, knowing that a lot of those pathways will just never be used. And those after age 2, generally, but not exactly universal to all areas of the cortex, that seems to be on average the halfway point, age 2. After that, we don't gain new neurons, we don't gain new connections between neurons. Instead, we are losing neurons, and we'll lose a ton of them. And that just goes to show how much redundancy and capability even a normal adult brain has. And then compare that to a child's brain. The development of the brain is one of the most amazing things that I've seen in psychology, to be honest. And the cell death sounds bad. Dying cells we think is going to lead to problems, and in some cases with adults it can. But for children it's a very good thing because it shows that their brain is adapting to things that they are seeing and dealing with in their environments. And then once it's got those pathways that it needs, it makes them faster through the myelinization process, turning those less efficient gray matter pathways into efficient and fast white matter pathways. Now we also have the idea of integration. There are plenty of examples we could use for many things, such as speaking in a conversation. There's one area of our cortex that lights up when we're understanding what someone's saying. There's another area of our cortex that lights up when we are producing words. And the idea is that as we develop, those two areas integrate and light up in faster or more rapid sequence, right? So they become more efficient. And I think about it with my 8-year-old. He basically, you can tell he's thinking about what he wants to say, but he can't think and talk at the same time. And so it becomes a very inefficient process. I know that his brain is going to keep maturing, and those areas for speech will continue to integrate, and he'll get better. Another couple examples would be with movement. So for example, coordinating motor movement, we have our motor cortex, and then we have our cerebellum. They both need to work. And if you think about a 1-year-old who's learning to walk, those pathways that link those two, the motor cortex and the cerebellum, aren't very efficient. To move forward 5 years, those pathways will be myelinated, and for example, your kid is trying to learn how to throw a baseball, those pathways will be much more bing, 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 in rapid sequence lighting up. And the final example would be, think about basic or primal fear. A noise happens and we get scared. That initial reaction takes place in our amygdala, in our old brain, and then a split second later, instead of like a cat, hears a loud noise and spazzes out and runs away and scratches something. Humans, hopefully we don't just beat someone up and go crazy because we heard a loud noise. No, we take that extra split second and then our frontal lobe kicks in, and ideally, as long as that stimulus didn't represent a real threat, our new brain, from an evolutionary perspective, our frontal lobe, will then communicate back with our amygdala and down-regulating that, or reducing the activity of the amygdala, and we can calm down, and we can experience having less fear through the process of integration. We also have the idea of differentiation, and this is when cortical neurons begin to specialize, which means that as we develop, especially from birth to adolescence, young adulthood, when we are doing something, watching a TV show, eating a food, playing a sport, the more we do it, the more intense, smaller areas of our brain light up. And so, for example, a 2-year-old looking at any sort of stimulus, let's say a teddy bear, will have a more undifferentiated, or we might say diffuse response. So a larger area of the brain has a less intense response. And as we get older, let's fast forward to 12 years old, see that same stimulus, whatever it is, but we can go with the teddy bear again, you'll see a much more differentiated response. So a much smaller area or areas of the cortex will light up, but they'll be burning a lot more glucose. They'll be much more intense. So again, a more differentiated response is a smaller part of the brain, the cortex, but a more intense and focused or energized response. I also want to introduce the idea of plasticity. I assume everybody has heard of this before. And if we think about, for example, a critical period, if, for example, your visual pathways are not formed in the first year of life, well, you can't go back and fix them later on, right? You're just not going to be able to see the world visually that somebody whose pathways did form within that critical period can see the world. Now, as we talked about before, critical periods are hugely important, but they're also interesting because they're relatively rare. Most of the stuff that we're going to talk about, even if it doesn't form exactly the way it should in the typical age range, for most things the brain is fairly plastic, which means that other areas can take on, for example, if there was a lesion or a tumor or brain damage. Of course, the idea is that plasticity decreases with age. So what we're setting up with this whole synaptic pruning cell death is that we're building up the brain to become more specialized in specific areas. And the more specialized it becomes, that means, well, when that area is damaged potentially, those other areas who've been specialized for other things are less able to take on those things. So plasticity for most things is fairly robust, but of course, when we have a more efficient brain through the synaptic death and then myelination of what's left process, we do see this as a downside. It's a necessary trade-off. Let's move away from the neuroscience a little bit and talk about some general patterns of development, first of all. And we saw this on the pictures of the, for example, prenatal development. This also continues into postnatal development. We have the cephalocaudal principle, which means head first, essentially. And so if we look at growth and also development, we see that infants have a huge head and a tiny body. And then as the years pass, basically the rate of growth in the head slows and the rate of growth in the rest of the body increases. And we can also track that in terms of motor skills. So basically, you learn to move your head around and lift it up and turn it side to side first. And then you go down, and then you learn to use your large muscle groups in your torso and chest and abdomen. And you learn to, for example, move your arms and maybe even pick things up with your fingers before you'll be able to do anything like that with your toes. So that's all the head down principle. And of course, we also have the, as you can see, proximodistal. This is growth and development from the inside out. And so basically, we look at muscle development. You're going to use those large inner muscle groups in the neck and the torso. And then you move out to the larger also large muscle groups of your biceps and your thighs to just roughly move your arms and legs. And then the last thing to develop is the fine motor skills in your fingers and toes for balance and manipulating objects. So again, from the inside out. I just introduced some of these in the last slide, but let's formally distinguish between two basic kinds of motor skill development. First of all, we have what are known as gross motor skills, and these are the ones that you have to typically learn for the most part first. And so once you get a baby that becomes a toddler, that then becomes an early childhood member, the gross motor skills have shown tons and tons of progress. So you start with basic things such as just learning how to walk, and then moving to toddlerhood where you're kicking a ball, you're jumping, you're squatting. And then also we have fine motor skills. And there is some development in the first year of fine motor skills, but it's very slow. And you can see fine motor skills involves things like grasping, balance, things like being on tiptoes, learning sign language, hand-eye coordination, even typing. You just can't expect a kid in their first year to do much of this. They might be able to understand signing, so you can sign to them, and they might understand what you're saying. They just won't be able to sign back very efficiently. And there's all sorts of products that supposedly can speed up motor skill development. I think most of those are probably not very much based on science. I think the thing to remember with motor skills is that they are very, very plastic. And so that if you know a child who is very delayed in terms of developing some sort of motor skill, that might be an indication that it's time to look to see if there's something wrong. But if there's nothing wrong with the brain or the body, those motor skills can be made up, and all that development can be made up very fast later on. So it's a classic example of some kids just take longer, and with motor skills, that's one of those you don't really have to worry about. It's important to know the general progression of gross motor development. And so what we see here is, first of all, you're just learning how to roll over and lift your head up. And again, it's a small body and a huge head, so it doesn't seem like much, but that's actually a big accomplishment. You've had to do a lot of muscle development just to get there. Then you start involving your arms and chest muscles. And then you see sitting up. So notice now we're getting to standing once we get toward the end of the first year. And notice they don't put crawling here. Some kids crawl, some babies crawl, some don't. It's not a necessarily normal or universal aspect of development. And again, these are just the typical age ranges, but if someone is delayed, and again, there's nothing physically or cognitively wrong or neurologically, then I wouldn't be worried. They can still catch up. One thing I want to emphasize at some points this semester are we talk about when should something happen? And, well, how do we know that? We can start with something like Erikson's theory of psychosocial development, and a theory is a good place to start, but at some point we need actual data. And where does the data come from? What is the best measure if you want to know where your kid falls relative to the average kid? Is your kid gifted in some attribute or falling behind? And I know you can't read all these, but this is the Denver Developmental Screening Test, and this is sort of the gold standard to know in terms of first 2 or 3 years motor, and we see gross motor, language, fine motor, and even some social skills. couple interesting patterns that we observe is that we don't see spontaneous smiling until the second month. Then we get into playing patty cake, for example, here, to much more complicated things like using a fork and a spoon, for example. We see all sorts of language stuff. And so first of all, they're basically not using words. They might be babbling starting around 6 months, but they turn to hear voices at a specific time. Then we start to see mamas and dadas, but it's mostly just babbling. Then you see evidence at close to the end of the first year that they're using mama and dada and some other basic words as actual words and understand the symbolic meaning behind them. So again, if somebody or you want to know whether your kid is above or below average, it doesn't really matter what their opinion is. There needs to be some sort of standardized test, and this is the one for early development that I would trust the most. We also see the idea of reflexes. Reflexes are very much, in the nature-nurture debate, these are nature. These are programs that are reflected in our genes, and in some cases adaptive to help us in our very first weeks after birth to survive. So of course we have what's known as the Moro reflex, which is support the baby from behind and then lean him back and the arms kick up. We also have the swimming reflex, put them in water, don't submerge them in water, just an inch or two will do in the bathtub. Grasping reflex, anything inside the palm should cause the palm to close. And then we have the walking reflex, which is take the 2-week-old baby, make sure they're supported, and then have their feet brush a surface, and the feet will start moving in basic walking motions. Now what's important is that these are reflexes. They haven't been learned through the environment at all. And what's also really interesting is that the swimming reflex and the walking reflex after a month or so will go away. And so to a parent, it might seem like you've lost something, right? A child is not responding in the way they used to. But what that also represents is you're moving away from just basic genetic programmed responses, and now you are setting the potential to have a higher order understanding of what does it mean to walk, what does it mean to have my feet on a surface. And so that's the start of a higher order processing of something that used to be very basic. Especially in the first, let's say, 8 months of a baby's life, they are what we call pre-linguistic. They don't really know language in any way that we do. And so how do you measure what they like, what they don't like? Obviously, crying responses and temper tantrums will give parents some indication. But we have this process known as habituation, which means you show something new to a baby and then measure how long it takes for that baby to get habituated or bored with it. So how long to habituation? And if you can guess which one does let's say a 4-month-old look at longest, they will look longest at this, the face, rather than just random attributes, and both of these more than the blank face. And this is an indication that, oh, well, they must have some sort of symbolic understanding that a face is meaningful. And they have no other way of telling us that, but here's one way to test it. And here we can see the mean degree of habituation. And in this case, one of the variables is how far are they willing to turn their heads to see that thing. Very unwilling to turn their heads very far for the blank face, and much more for anything with something in it, but especially one that represents a real face. We can also measure all sorts of other, besides head rotation, ways that babies can tell us things. One is measuring their heart rate, measuring eye fixation, where are they looking, the rate of how fast they're sucking, kind of like Maggie Simpson. When she's excited, she sucks her pacifier very quickly. That's actually kind of scientifically accurate. We also see that a 4-month-old might look at this object, which is basically physically impossible, longer, or it takes them longer to become habituated to this, which also shows that they have some basic expectations, even at this young prelingual age, about what their world should look like and which edges should be where for a three-dimensional object. And a general pattern here with this kind of research is that babies, the more creative research that we do, we typically get more evidence that babies are understanding more and have more symbolic understanding of the world around them than we'd previously given them credit for scientifically, because we haven't been able to measure it. You have to come up with these very creative research paradigms. Here's a measure of eye fixation. I think this one is great because if you've ever held a 1-month-old, you're wondering, why are they looking above my head so much? Well, partially because their visual acuity isn't very good and they can't see the eyes distinctly very well, but also because they don't know at 1-month-old that symbolically, to understand somebody's mood and feelings, the eyes and the mouth are where to look. Contrast that to just 2 months later, and we see a huge difference. There's still some stray above the head shots, but lots more focusing on the mouth and the eyes, and not just spending time there, but going back and forth between these. This is another example of perhaps integration. They're integrating their understanding of how to read a human face. Again, this is prelingual research, but trying to test basic expectations of what's possible, what's not possible. And so let's say we'll have a 6-month-old, and you'll have to do multiple trials because sometimes they look away. But assuming you got them paying attention for all these 3 rounds, you show them the practice round, right, where the car goes straight through onto the line. Possible event, the toy is behind the line. Will they understand that this is possible compared to this one, which shouldn't be possible, which if the mouse is here, the car still goes straight through. And sure enough, they will spend more time looking at the impossible outcome, suggesting once again that they have a greater understanding of how things should work. And again, an understanding that's there despite not having a language to represent it. So one thing I wanted to distinguish between is the basic sensation versus perception. And basically, sensation is all the inputs that are coming into our brains to be processed. Everything we feel, everything we see, everything we hear. Perception is the way we organize. Oftentimes we'll see this term schema associated with Piaget. And so we see something new. Do we even have a label for it? Or is it just, this is a new thing, I don't understand what this is. So perception is a greater and greater understanding and organizing of the world. And so going back to the impossible event, the baby is just not passively watching a toy and a moving object. The baby is perceiving what is possible and not possible in a situation. Now, we also have to think about babies perceive their world in a different way than you or I do, because as adults, we are visually dominant. We rely on our eyes more than our other senses. That's not the case with babies. They still have to make that switch, which is going to come a little bit later on. And so a lot of their understanding of the world is through how objects feel and how objects interact with them. So think about if your phone is pulsating. So toys that respond to certain kinds of manipulation, some with buzzes, some with pulses, others with lighting up. This is the idea of haptic perception. And it's important that we can obviously learn through haptic perception, and technology is basically helping us do that, but babies are naturally doing that more than we would. And so any sort of toys and objects that involve haptic perception, a baby's just going to get more out of. They're going to be more interested in it because that lines more with what they are doing and the basic processes they're using. couple things to think about as we wrap up. What is the basic progress in terms of sensation and perception? So for example, audition. Well, babies can hear things before being born. They can learn what their mother's voice sounds like and distinguish between an anxious state versus a relaxed state. So audition has already taken place to some extent before birth, and even smell and taste. So babies already have taste preferences, and they prefer things such as sweet like bananas and things that they've already been exposed to even before birth. Now vision is the one where it's had hardly any development before birth. There's no stimulus, the eyes aren't open, it's just not ready to come online really until birth. And so we see depth perception has a long way to go after birth, and basic visual acuity and color vision also needs time to develop. So first of all, right after birth, they're 2600, so they're basically functionally blind. And then we go to 2100 in 6 months, right? So that's far away from 20/20 adult vision, but it's a huge progression from birth. And here we see a basic example. So this might be, let's say, 2 months, 4 months, 6 months, a year. So you can see the drastic development and how fast visual perception develops relative to where it started. And of course, then we look at those brain pathways that process visual information. By the time they hit 1 year, many of them will already be myelinated, changed from gray matter to white matter. So the brain development has represented what we see in terms of their visual progress. Depth perception is interesting for humans because most animals after birth, such as horses and goats, they, from an evolutionary perspective, have to be able to not basically walk off a mountain. So the depth perception has to be there, and when you put them on this visual cliff, right, they won't actually fall because there's plexiglass here. The newborn goats will not venture off the visual cliff because they have depth perception, whereas babies, oftentimes about 4 months where they start to learn to crawl or can at least roll off, they don't have depth perception. So if parents aren't careful, they'll just roll right off a counter or a couch and potentially hurt themselves. So there's this weird asynchrony with humans where they are able to move before they're able to perceive depth and danger. So that's one of those potentially dangerous periods that once they start crawling, don't just assume that as a parent or caretaker that they're going to avoid falling. And finally, as we wrap up, these won't be an exam. I had covered these in a previous version of the class, but they do represent some different perspectives in how to understand human development. And again, these aren't for the exam, so I'll just go over these quickly. Basically, the ecological theory of perception is ecologically, what's the simplest way to explain the progression and the timing? And the whole idea here is you're looking for human universals. This is the critical period approach, and it's shifting from bottom-up understanding to organizing your environment, to knowing what's dangerous, knowing what's safe, and also showing what we observe, that senses show asynchronous development. You're developing hearing and audition before birth, and then after birth is pretty much visual development that's dominating, and the audition continues to develop, but at a slower rate than the vision. They don't develop all in sequence or in the same rate. So again, this is very much focused on the research, on the timing, on human universals to successful and normal development. But on the other hand, we have the dynamic systems theory. And this is saying order is largely the same as the ecological, but we have different environments that explain observed differences. So for example, going back to muscle development, we have what is normal timing for when a baby should start to learn to walk. We have other historically indigenous cultures that basically swaddle their babies and carry them around for the first 2 years of life, so they don't get out to walk. Dynamic systems theory says, well, we see a behavioral delay in terms of motor development for these groups, but it's all a product of the environment. And once you remove the swaddling, after those first 2 years, the child can then learn to walk very rapidly. Also, we have this idea of, with dynamic systems theory, a more fine-grained look at the context of the environment. And this is more similar to the idea of Vygotsky's zone of proximal development. It's not just, is the sensory development there or not, and if not, is there a problem? It is much more nuanced, and how far on the journey are they for this specific thing? So, long-winded approach, and again, this won't be on the exam. Hope we wrap things up nicely, and you got something that you'd like to discuss, either here on the chat boards or on the message boards. Thanks for hanging in there for this hour and 20-minute version of a narrated slideshow. Thank you.